Let us talk about on the general process of how biobutanol is produced. Here we can see a simple flowchart whereby firstly, biobutanol is mainly derived from the fermentation of sugars in organic feedstocks. Butanol is a byproduct of this process, in addition to ethanol and acetone. The process, known as acetone-butanol ethanol fermentation, which uses the microbial species Clostridium acetobutylicum. Biobutanol, on the other hand, has the potential to be applied as future transportation fuels, but then it is still not economically feasible due to several constraints. Therefore, this brings to our problem statement, whereby the main problem rise from biobutanol production is its toxicity. For instance, at high concentration of butanol, it causes inhibition towards the Clostridium species, which leads to denaturation. Not only that, high cost is needed for product recovery. In the same context, our solutions are more derived towards engineering-based. We as engineers, we want something that is cost-effective, easy to handle, and provides better productivity. All of this can be achieved through adaptive laboratory evolution, AL method, whereby it's actually a technique of improving the strain through serial cultivation with different pressure until it can be adapted to end goals we want. In order to achieve our goal, AL method was applied to our locally isolated Clostridium acetobutylicum YM1 strain using serial cultivation to enhance its tolerance under specific selective pressures. For instance, in our study, we emphasized on improving the strain tolerance level by subjecting to high biobutanol concentration that starts from 15 gram per liter and end at 23 gram per liter. Next, we will briefly discuss on our methodology. We start the adaptation with 15 gram per liter because we found that this is the inhibitory butanol concentration for our strain. Once the strain has acquired the strength to sustain at that particular concentration, then we move up to next concentration. This is continued until the strain reached its limit. Then, once the best adapted strain was produced, it was scaled up in a 2-liter bioreactor using optimized TYA medium to assess its feasibility for industrial application. So now if we look into our result, we can see the success in adopting the ALE approach through the improvement of biobutanol production yield and productivity in adapted strain, but we can also see that the improvement is not very apparent. Therefore, we came up with a novel method that can enhance serial cultivation method during ALE, which is called activation approach. So how does this approach helpful? This method prevents overexhaustion of the strain and also to promote longevity of the strain to tolerate higher butanol concentration. As a result, we can see that the adapted strain it can sustain up to 22 gram per liter of biobutanol and produce 11.67 gram per liter of biobutanol, in contrast with wild strain with negligible amount. This adapted strain of 22 gram per liter was further selected for closer comparison with wild strain. Here we can see that even when butanol was added to the medium, the adapted strain can produce high biobutanol without impacting the viable cell. In this case, butanol was not added to the medium, and the biobutanol produced was significant for a lab scale result, which was up to 21.26 gram per liter of biobutanol produced. On the other hand, scaling up strategy revealed high amount of biobutanol produced that further highlights its feasibility for industrial application for biobutanol production. As for invention advantages, our study has managed to enhance biobutanol production with significant improvement from lab scale to scaling up, and also butanol tolerance, in which there is 7 increment, which is by far the highest compared to previous study that only has 1 to 2 increment improvement. In terms of cost estimation, it is also proven how cost-effective employing ALE strategy in contrast to genetic-based strategy. In terms of novelty, our adapted strain, the first Clostridium species in the world, that could tolerate butanol concentration up to 22 gram per liter. We also have our own method produced in patent under my IPO. Not only that, our research achievement so far, we had won 1 gold and silver medal, have produced a thesis, and also have collaboration with industry, which is Free, the seed company, whereby they are interested in using our adapted strain and utilize it with rice straw waste for biobutanol production. Here are some of our published papers that highlight on the scope of biomass and renewable energy, specifically in biofuel production. These are few major papers that we have referred in completing this research and slide presentation. That comes to the end of our presentation. Thank you so much for your attention.